All right, so moving on, we'll talk about the proximal convoluted tubule. So this is just an overview of the processes that takes place. All right, we're going to look more closely at those processes, but in the following slides, we're pretty much going to paint a big picture of what happens to each part of the tubular structures. So in the proximal convoluted tubule, again, this first region, this first twisted or convoluted region right after the glomerulus, what happens here is the absorption of organic nutrients, right? You have amino acids being absorbed here, ions such as sodium and chloride, and protein. Now, protein should not be in the filtrate, right? Protein should not be in the filtrate based upon the size of the glomerulus. We'll talk more about this. There should be no protein in the filtrate. But if there is damage, protein will be reabsorbed in the proximal convoluted tubule. Just because the body does not want to get rid of protein, protein is such a vital macromolecule that we reabsorb it right at the start, so we're going to reabsorb it in the proximal convoluted tubule. Also reabsorbed here is a lot of the water. Okay, so this is important in regulating blood volume. So as we filter all of that volume out, we do not want to lose all of that water. Think about you losing all of that filtrate, right? So if all of the blood that you filtered was lost, you'd be severely dehydrated on a daily basis. So a lot of that water has to be reabsorbed to help maintain blood volume. The capillaries in the proximal convoluted tubule are associated with peritubular capillaries. So we talked about this. Remember those two important vessels I told you to remember? The peritubular capillaries is going to be closely associated with the proximal convoluted tubule. So what does that mean? What does that mean? Having those veins closely associated with the proximal convoluted tubule, what's allowed to happen here? If I'm hearing it. What's allowed to happen from the proximal convoluted tubule across into the peritubular capillaries? Reabsorption, right? So we're allowed to reabsorb water, we're allowed to reabsorb those important things that I told you we needed, such as glucose, such as amino acids, any protein that may have spilled into the urine.
So it says that they're called the peritubular capillaries?
Yep.
But you said they're associated.
They're associated with the proximal convoluted tubule. They're called, the tubule is called the PCT.
Oh, okay, all right.
And then those veins, those capillaries wrap very closely around the PCT. Okay. All right, moving right along, let's look at the descending portion of the nephron loop or the loop of Henle. So the descending part of the nephron loop is going to be mainly responsible for water reabsorption. Right? So this is the boring part of the nephron. Only water is absorbed here, no salts, no other ions are moving across this part of the tubule, only water. And the movement of water here is very important in establishing a gradient, which we're going to talk about. So in the descending limb of the loop of Henle, only the movement of water. Okay? And again, that water is going from the filtrate, which sits in the tubule, out into the blood vessel. Now it's going to be going to a different type of capillary. That second capillary that I told you is important, the vasorecta. Okay, so just to recap here, the proximal convoluted tubule is intimately associated with the peritubular capillaries, but the loop of Henle is intimately associated with the vasorecta. And so when reabsorption of all that water takes place from the filtrate in the loop of Henle, it's going to be reabsorbed into the vasorecta, and it's going to, again, help to reestablish, help to maintain blood volume, preventing us from losing all of that water. Okay? In the ascending portion of the loop of Henle, the opposite takes place. So here we only have the movement of salts, sodium, chloride. We have no movement of water. All right, that's going to be very important as we move on. So the descending part of the loop of Henle, only the movement of water. Ascending limb of the loop of the Henle, only the movement of salts, only the movement of sodium and other ions. Right? Any questions so far? Okay. Looking at the distal convoluted tubule. So this is where another process takes place. This is called secretion. And if absorption or reabsorption, sorry, so let me make that distinction here. There's no such thing as absorption in the kidney. It's always reabsorption. Why? Because we have a filtrate. We filtered the blood, and so we have a filtrate. And anything that we're going to move across that tubule is going to be reabsorbed, not absorbed, right? But looking at secretion, if secretion is the opposite of reabsorption, where do we think the movement of these substances are taking place to? What do you think secretion is? Talk about reabsorption. Going back in. So it's going to be going from the blood, going back into the filtrate, into that tubular secretion. So this is basically things the body wants to get rid of. Drugs, excess sodium, excess calcium, any sort of medications that we're on. This is the part of the collecting system. This is the part of the kidney, the tubular structures of the kidney, where it's basically now or never. You have to get rid of anything that you don't want, and that happens to be excess ions. Any drugs that are present in the system can be secreted to be eliminated at this point. Very little reabsorption of water takes place here, and that depends on the hydration status of this person. And finally, the collecting duct. This is the final part of the tubular structure. After this, we basically have urine, and that's going to be expelled to the renal pelvis. So in the collecting duct, we're draining the fluid from the distal convoluted tubules. And at this point, we're really making any last-minute adjustments. And this is what we call regulated fine-tuning. This is regulated. And regulated means that based upon external factors, factors like your hydration status, factors like your blood volume status, factors like your fed versus fasting status. So basically, depending on the state of your body, we can either reabsorb water here or we cannot, right? So that is regulated in that way. But at the collecting duct is the final place to make adjustments before this is formed as urine. So let's look again at this overview image, which is sort of comparing all of those parts that we talked about. So we had the proximal convoluted tubule, right? And this has some important structures we're going to point out as we move on. This has lots of mitochondria and microvilli. That's going to be very important to the function of the proximal convoluted tubule. We'll talk about why. We have the distal convoluted tubule. Again, very similar to the proximal convoluted tubule, but here we're looking more for secretion of anything that's excess in the body, excess ions, drugs, toxins, etc. Those can be released into the urine to be eliminated. We have the renal corpuscle. So remember I told you the renal corpuscle is the glomerulus plus the Bowman's capsule or the glomerular capsule. Those two structures forms your renal corpuscle. And basically this is where the blood from the vessels is being filtered, and basically we have a filtrate. Now let me just backtrack here. Some students often get confused when I say filtered. Filtered means that we're going to pass this blood through some sort of sieve or some sort of semi-permeable membrane. And semi-permeable means that some things are going to go through and some things are not. That's what a filtrate is. So in the case of urine, when we filter the blood, what are we obviously not taking? What are we obviously trying to leave in the blood? What do we not want in the urine? Plasma. Hemoglobin. So red blood cell, that's one. What else did I tell you was never in the blood? Proteins. Those are size-dependent structures that are not going to pass into that filtrate. So you should not have blood in the urine ever, unless there's injury or damage. You should not have protein in the urine ever, again, unless there's injury or some sort of damage. All right, so that's what I mean by filter or filtrate. So what's left in the urinary space is now our filtrate, absent proteins, absent blood, everything else, right? And finally, we looked at the two main collecting parts here, which are the connecting tubules or the collecting duct. Right? These two structures are synonymous. They basically have the same function. Reabsorption of most of the water here, last minute fine tuning depending on hydration status. So if you're well hydrated, we're gonna lose a little bit of water here. You can stand to lose some water. If you're severely dehydrated, we're gonna hold on to any bit of water that we can, and almost no water is gonna be secreted here. Okay, so we have that last minute fine tuning of ions and of water in the collecting duct and the connecting tubules. Okay, let's look at the juxtaglomerular complex. The juxtaglomerular complex. Now, this is a series of 3 different structures that are responsible for basically fine-tuning or regulating the glomerular filtrate. So the macula densa, the juxtaglomerular cells, and the mesangial cells, also called the extramesangial cells, these are basically sort of testing the blood that's flowing past them. So think about you actually sort of tasting a pot or sort of testing the weather. That's what these structures are doing. They're sensing, they're sort of sensors that are picking up on the pressure of the blood that's flowing out the efferent arterioles. All right, so just to remind ourselves here, this is the afferent arteriole. This is bringing blood to the glomerulus to be filtered. Blood is filtered across the glomerulus here, and now all of it does not get filtered. The remainder of it flows back out of the efferent arterioles. Okay, so we have blood coming in here, being filtered or squeezed through the sieve to make a filtrate, and then the rest of it goes out the efferent arterioles. Now, when the remainder of that blood goes out the efferent arterioles, it comes into contact with these 3 structures that we just mentioned. And these 3 structures are basically testing the temperature of this blood. Is it flowing too fast? Is it flowing too slow? Is it under enough pressure? Do I need to fine tune something about the afferent arterioles? Do I need to constrict those vessels? Do I need to relax them? So basically these 3 cells are testing or taking or monitoring or sensing the flow of blood in the efferent arterioles, and then it's making fine-tune adjustments. We're going to talk about how those fine-tune adjustments come about later on down. So as such, these 3 structures together, which forms an endocrine organ, essentially, they are responsible for producing renin. Does anyone know what renin is? Anyone remember what renin is? It's important in maintaining blood pressure. So renin is that hormone. We'll talk about it. It's part of the RAS system, the renin, angiotensin aldosterone system, and that basically fine-tunes blood pressure. So it maintains blood pressure. Maintains blood pressure and helps to keep perfusion. Right, we'll talk more about this as we move on. The second function here, the second endocrine function is going to be the secretion of erythropoietin, and erythropoietin is responsible for new red blood cell production. Okay. Let's move on to our third objective, which is to look at the remainder of the collecting structures for urine. So that's the ureters, the bladder, and then the urethra. Okay. So the ureters, we talked about these in class. These are hollow muscular tubes that are leading from the venal pelvis down to the trigone of the bladder. They're bilateral, they're pulsatory, meaning that they're not just tubes sitting there, they're actually made of smooth muscle. So they're sort of pulsing that flow of urine along. And they're exiting the kidney at the hilum and entering the bladder again at the urethral orifice of the trigone, those 2 openings on either side. They enter the bladder at the posterior inferior side, so basically the underside or the base of the bladder is where they enter. And this is really important for creating that one-way valve that I talked about. So if they were entering the bladder on top, there would be a clear opening where if the bladder was full, you would have some urine going back into the ureters, right? So more there. So urine going back into the ureters. You don't have that because of the one-way valve that's been formed as the ureters open into the underside of the bladder. All right, so that prevents any urine from being collapsed back into the ureters. The histology of the ureters, we're not going to get too deep here, but again, there's 3 layers to not only the ureters, but the bladder. Much like most hollow organs, like stomach and uterus, there are 3 layers here. The inner mucosa, which is lined by epithelium, transitional epithelium to be exact. The middle muscular layer, which is basically the meaty layer of the ureters. And then we have the adventitia, which is the outer fibrous connective tissue capsule. Okay. Looking at the bladder. So the bladder is going to be located quite differently for males and females. The bladder is going to be in between the rectum and the pubis in the case of males. Right, so we pointed this out earlier. We got our bladder here in males. This is the pubis. This is the rectum. This is the prostate. This is the penis. All right, this is the pelvic floor, the floor of the pelvic cavity. And this is our bladder. Okay. For females or for women, we have the bladder in between the uterus and the pubis. All right, so think about, again, a pregnant woman having a very distended uterus, pretty much squashing her bladder, and she's going to have to go to the bathroom quite often, right, because of that pressure coming from the uterus. I also want to point out here that there's quite a short distance for the urethra. So women have a shorter urethra, it's about 4 centimeters long, whereas men have a very long urethra. It has 3 different parts that we talked about. So it's very long, much longer than women, and this makes women more susceptible to urinary tract infections. Okay. Moving right along, let's talk about, actually we're going to skip over these ligaments, they're not very important. Let's talk about the detrusor muscle. So that middle muscular layer of the bladder is called the detrusor. And again, we talked about the trigone, which is that triangular region of the base of the bladder where the ureteral orifice is open. Now at the neck of the bladder, we have the internal urethral sphincter. And the sphincter is basically a patch of smooth muscle that is under the control of the autonomic nervous system. So that patch of smooth muscle under the control of the autonomic nervous system is basically constricting when the bladder is full to keep it closed, and it's going to relax to empty the bladder when that comes about. Now, the urethra continues out into the urogenital diaphragm, which is basically the pelvic floor, and it has the external urethral sphincter. So out in the pelvic floor, we have the external urethral sphincter, and this sphincter is under our voluntary control. So when you think about going to the bathroom and you're ready to void, this is the muscle that you're relaxing in order to void your bladder, in order to void urine. You cannot control the internal urethral sphincter, but you can control your external urethral sphincter. So this is going to show some signs of deterioration with age. So we're going to lose control over this sphincter as we age. This is where we have that urinary incontinence with age, as well as some spinal cord injuries. And again, if we're, this is not developed, so if we're in like toddler or pre-toddler ages, we do not have as much control over this muscle, the sphincter right there. Okay. All right. Any questions so far? Any questions? All right, so here we're just sort of reminding ourselves, we mentioned that the female urethra is about 4 cm, it's shorter, it's closer to the vagina, so it has more propensity to have infections, whereas males is about 18 to 20 centimeters in length, and it has those several different regions that we talked about. The prostatic part of the urethra, which goes to the prostate, the membranous part of the urethra, which is in the floor of the pelvic cavity, and then the spongy urethra, which actually traverses or goes through the corpus spongiosum. All right, so this is just an image to help us compare the bladder in males to the bladder in females. Okay. Now we can see it has much more room, much more play or distensibility in males than it does in females. This is another reason why women get more UTIs. We have less sort of extensibility of our bladder because of the uterus, especially around the menstrual period or the menstrual time when the uterus may be distended. And so we have that susceptibility to infections. Let's talk lastly about micturition. So then micturition is just a fancy word for peeing or voiding. And the urge to urinate first comes when the bladder fills to about 200 milliliters. Right now, that's not a lot compared to the maximum that the bladder can hold. So basically, we feel the urge to pee when the bladder is not even halfway full. Okay. So at that point, we have the opportunity, or we have the control over the external urethral sphincter. We can open it and we can void the bladder at that point. If that does not take place, the bladder will continue to fill. As we continue to make urine, the bladder will fill. So it will go from 200 mL to 300 mL to 400 mL to 500 mL. Between 500 and 800 mL, we have no control over the external urethral sphincter. When I say that, I mean we cannot try to keep it closed any longer. This is where we have to go to the bathroom, right? This is that point where we lose the control of that external sphincter. It's going to open whether we want it to or not. And that's synonymous to wetting your hands, right? Okay. And then lastly, we'll touch on some age-related problems here that are seen as the kidneys age. So the nephron decrease in number. Also, what has been shown is that the length of the loop of Henle decreases as well. We'll talk about the importance of